Hello everyone. In this video session of forensic medicine, I would like to discuss about bloodstains and its importance. See, whenever a crime has happened, a murder or a homicide, the bloodstains serve as one of the most important evidence which will help the police or the civil surgeon in carrying out the case and delivering justice. So, remember, how will you collect the blood stains? Remember, we will always use a piece of clean white cloth or a white filter paper or a cotton swab. What is the solvent which is most commonly used? 10% solution of potassium cyanide or 10% solution of glycerine. I'm not saying each and every line, only what are the salient points which are needed. I'm just explaining. How will you preserve the blood stain? Remember, it is allowed to dry at room temperature and no extra heat should be used because it will cause deterioration of the stain. There are certain substances which resemble blood stain. For example, rust stain, synthetic dye stains, mineral stains, vegetable stains. Okay, coming to examination of blood stains. Blood stains can be examined under different headings or there are different types of examination. Number one, general examination, chemical examination, microscopic examination, microchemical examination, spectroscopic examination, okay, and serological examination. So these are the different ways of examination of blood stains. This is extremely important because in different situations we will be using different types of examination. So coming into what do you mean by general examination? The stains which are found at the scene of the crime. For example, as you can see in this picture, just focus on this picture, you can see spurting. So the spurting of the blood stain indicate the direction. In second, you can see by seeing these drops of blood, you can see that the blood has fallen vertically, and you can see the various sizes of increasing sizes of the blood stain, which increase the drops of blood from the various height. As the height increases, the size of the drop also increases. This is actually called as general examination. Once that is done, let us go for the chemical examination. There are specific names for the test we use for chemical examination of the blood stain. Just remember what is the name of the test and what is the reagent. Just 1 or 2 points, that's enough. Number one is called as the benzidine test, which is actually absolute now. Okay, that means we are not using this test now. Here we will be using benzidine reagent, and if the blood is present, a dark blue color will be produced. That is enough. Second test is called phenolphthalein test, also called as Castile-Mayer test, using phenolphthalein reagent. Okay, and here if the blood is present, a pink or purple color will develop immediately. So these are the two important chemical test, that is benzidine test and phenolphthalein test, also called as Castile-Mayer test. Castile-Mayer test is the name which is more commonly given in your exams. Coming into microscopic examination, all the stains we will scrap out, we will put in the glass slide, we will mount it, observe under the microscope, we will see RBC, that will confirm if it is a blood stain. How will RBC looks like? Biconcave, non-nucleated disc. Coming into microchemical test, that means based on the property of the heme part of the hemoglobin, okay, which will have some tendency to form characteristic colored crystals when used with certain reagents. Here we have some certain name of the test. Out of that, the two most important one is called Hemin crystal test, also called Teichmann test, and hemochromogen test, also called as Takayama test. Okay, there is no need for you to know about the procedure of the test. Just remember Hemin crystal test, also called as Teichmann test. The blood stain or the Hemin crystals will be brownish black rhombic crystals. So remember, Teichmann test is brownish black rhombic crystals. Whereas in case of Takayama test, you will see pink feathery crystals of hematite. Okay, what is it? Pink feathery crystals of hematite. That is the 2 points you have to remember. So Teichmann test, brownish black rhombic crystal. Hemochromogen test, it is a feathery pink crystals of hemochromogen. Then coming into spectroscopic examination. Number 1 is oxyhemoglobin method, reduced hemoglobin method. Okay, and so these are an acid hematine, alkali hematine method, methemoglobin method. Okay, so these are the various tests you would have already studied in your physiology. Okay, in hemoglobin estimation, you would have already studied the names of this test. Just remember the name. And the last one is called as serological examination. That means if the blood stain is from the human beings or the species, it helps to identify the species, whether it is from any animals, whether it is from the human beings, everything. So that we will use a technique called as precipitin test. Precipitin test is an immunological method. It is a serological examination. Okay, fine. Just remember the name. That's enough. And next is called antiglobulin consumption test, gel diffusion test, and precipitation electrophoresis. Okay, yes. So these are the different importance. Okay, these are the different serological test. What is the medico-legal importance of blood group? We have different types of blood grouping: ABO blood grouping, RH blood grouping, Kell, Duffy. Okay, Lewis. Lot of different types of blood groupings. What is the medico-legal importance? That is the one thing which we have to discuss. Number 1. A blood group antigen cannot appear in a child unless present in one or other parent, father, mother. In either of them, the antigen should be present. Then only the baby or the child will get the blood group antigen. If an individual is homozygous for a blood group factor, it must appear in the blood of all his children. If a child is homozygous for a blood group factor, the gene must have been inherited by it from each of its parents. And the blood group characters are peculiar to the individual and they are unchanged throughout life. That means when a child is born as B positive, he will be throughout life, he will be B positive. So these are the 4 important medico-legal aspects of blood groups. Okay. So just remember the name of the different blood group system. ABO system, that is the most common. Then we have the RH group, the RH system. Okay, the RH stands for rhesus monkey. Okay, because this antigen has been isolated for the first time from the rhesus monkey. And what are the different antigens which is present in the RH system? It is capital C, small c, capital D, small d, capital E, and small e. Okay, fine. Out of that, the most important one is the capital D, small d system. Fine. Then we have the KIT system, Duffy system. Okay, the MNS system, Lewis system. These are the different names. Just remember the name. That's it. Okay. So, medico-legal application of blood groups. This is a very, very important question. It's a 5-star question. Okay, it's a VVIP question that you have to focus on. Medico-legal application of blood groups. Number 1, disputed paternity. For example, the question of disputed paternity arises in court in the following condition. What are the conditions? Suppose when a child is born in a lawful marriage, but the husband denies that he is the father of the child. Or when a child is born out of a lawful marriage and the mother accuses a certain man of being father of the child while the man denies the accusation. Third, when a woman pretends pregnancy and delivers and obtains a child claiming it as her own. Okay, that is called fictitious child. What is the name of that? It's called as fictitious child. Fine. So these are the various conditions in which there is a disputed paternity will occur. So in that case, we will do blood grouping system. Second thing, disputed maternity. The same thing happens here. Okay, what I have told you, disputed paternity, the same thing. And crimes, okay, blood stains. Okay, the victim accused can be matched off. Then see the identity. Then for the cause of death. So just remember identity, cause of death, disputed paternity, disputed maternity, and crime. So these are the 5 different medico-legal importance of the various blood group system. Next comes another very important question that is called as DNA fingerprinting. It was first developed by Dr. Alec Jeffreys. Dr. Alec Jeffrey. It is a technique which involves chemically dividing the DNA into fragments. DNA will be converted into fragments, various fragments, and finally, each of us have a unique pattern. Remember, identical twins have same type of DNA fingerprint. So how will you differentiate identical twins? Yes, very good. That is mainly by our fingerprinting. Fingerprinting is unique to individual. Even in identical twins, the fingerprint is different. Okay. So using gel electrophoresis, this fragments will be arranged, okay, that is based on the positivity and negativity, and then we will be decoding a particular profile, a unique profile of DNA fragments for the individual. That is actually called as DNA fingerprinting, which was first discovered by Dr. Alex Jeffreys. So what are the different methods in which, and we have something called as, okay, before that we have something called as the regions which contain repetitive DNA and which demonstrate hypervariability from person to person, that is actually called as satellite DNA, and these variants are called as VNTR, or the variable number of tandem repeats, also called as mini satellites. They are nothing else but you can see almost only some 0.1% codes for proteins. The rest everything is just repeats of the same fragments, okay, and these repeats can be further identified by two important techniques called as RFLP, that is restriction fragment length polymorphism, and next is called as PCR, polymerase chain reaction. What do you mean by RFLP method? The DNA will be extracted from the body fluid or tissue, and all the samples will be frozen at minus 20 degrees Celsius. Fine, and the DNA separation, which will include the disruption of the cells, the DNA will be isolated, okay, and finally we will use some certain protein to remove the RNA and polysaccharide, and finally the isolated DNA is quantitated by ultraviolet spectrophotometry, and the DNA will be completely digested with the help of some group of enzyme called as restriction endonucleases. Okay, fine. And this restriction endonucleases will cut the DNA at some specific fragments, and these fragments are called as restriction fragment length polymorphisms. Okay, and remember, this restriction fragment lengths are produced because of some tandem repeats, which is called as VNTR, or the variable number of tandem repeats. That means same sequences of the DNA are repeated again and again and again, which makes some billions of base pairs in the DNA. Fine, okay. Just remember this is the picture you have to remember. Number one, biological material, we have to isolate nucleus, isolate and purify the DNA. The DNA will be digested with the help of restriction endonucleases. Then DNA fragments are separated by gel electrophoresis, and we will be transferring it to a nylon membrane. For DNA it is called as southern blotting. For RNA it is called as northern blotting. For proteins it is called as western blotting. And hybridization of labeled DNA, then we will do hybridization using radio labeled probes. Then we will wash the membrane, and we will do something called as autoradiography, and finally it will be visualized. And next is actually called as polymerase chain reaction. You should know what are the different steps of polymerase chain reaction. Okay, that is very, very important. As far as polymerase chain reaction is concerned, suppose we have only very small amount of DNA is available. What we will do, we will amplify it multiple times, and then we will do DNA profiling. Okay, yes. So this is done first with the help of primers. The primers will act as the starting point for the DNA duplication process, and first the initial temperature will be 94 degrees Celsius, then it will be 72 degrees Celsius, and finally it will be around some 54 degrees Celsius. Okay, these are the three different temperatures of polymerase chain reaction. As you all know, polymerase chain reaction was discovered by Carrie Mullis, okay, for which she was awarded Nobel Prize. And remember, we will use a thermostable DNA polymerase called as Thermus aquaticus polymerase, or the TAC polymerase, okay, which will help in synthesis of the DNA fragments. That is called as amplification, and then there will be extension of the primer. Okay, there will be extension of the primer, and as a result, multiple, that is 30-40 cycles will be repeated, and which will help produce millions of copies of the DNA. Fine, okay. What are the importance of doing all these things in case of murder? Okay, sexual crimes, victim and accused, paternity dispute, maternity dispute, exchange of newborn in the hospital, identification of mutilated remains in case of accidents, wars, disasters, bomb blast, okay, and identification of bodies in exhumation cases, for species identification, and all cases of biological identification. These are the different medico-legal applications, like how I have told what are the medico-legal applications of blood group system. Similarly, all these which I have mentioned, these are the medico-legal applications of this RFLP and polymerase chain reaction. So in this chapter, the main questions that you have to focus is polymerase chain reaction, the three important steps of polymerase chain reaction, what are the three different temperatures of polymerase chain reaction, DNA fingerprinting, remember Alex Jeffreys, remember RFLP, remember variable number of tandem repeats. Then you should know what is the medico-legal importance of this DNA fingerprinting, medico-legal importance of blood grouping system. Okay, see, remember this is very, very important high yield slide. What are the different steps of restriction fragment length polymorphism? Just remember this picture and write it in the form of a flowchart. And next, another most important point is what are the medical importance of the blood group system and what are the different types of examination of the blood stage. General examination, chemical examination, microscopic examination, microchemical examination, spectroscopic examination, and finally is the serological examination. Just remember the Hickman test, then Takayama test you have to remember, Benzidine test you have to remember, Castelmeyer test you have to remember, and then is what becomes the Precipitin test, okay, and the spectroscopic method like oxyhemoglobin, methemoglobin, acid and alkali hematine methods, okay. So these are the various important things that you have to remember in this chapter, and I think with this we have come to the end of this chapter. So thanking you for listening patiently to this video, and in the subsequent videos I will be discussing the rest of the topics like sexual offences, rape, incest, okay, natural and unnatural sexual offences, and forensic science laboratory and forensic psychiatry. Thank you.